Hi everyone, my name is Molly Field and I did my creature feature video on the process of phagocytosis in cauliflower coral and in the starlet sea anemone. The Pachylophora damicornis is also known as the cauliflower coral. They are found in the Pacific Islands and they typically live in communities between 1 to 27 meters deep, and they're found in shallow reefs that are exposed to strong wave action. Cauliflower coral face a number of threats, including ocean acidification, ocean warming, habitat degradation, and disease. They are currently listed as an endangered species as of February 2021. The corals most commonly have a cream color, like on the bottom left picture, but they can also be green or pink. The Nematostella victensis, or starlet sea anemone, is a tiny flower-like invertebrate. They are found in shallow, salty lagoons along the coast of North America and part of the United Kingdom. They feed on small mollusks and crustaceans by using their venom-filled tentacles to pull prey towards their mouths. They are the first species known to grow new limbs as a response to food consumption. The more food they eat, the more tentacles will grow. The image on the bottom shows the progression of the anemone growing more tentacles. Each anemone starts out with 4 buds around their mouth that will be their first 4 tentacles. The growth of additional tentacles is dependent on food availability. The starlet sea anemone is hard to find and is endangered due to climate change. Phagocytosis is a process where immune cells destroy foreign pathogens. Basically, phagocytes engulf or ingest other cells or particles. This process is common in immune system cells to destroy bacteria and viruses. This picture illustrates the process of phagocytosis. In August of 2021, it was discovered that both cauliflower corals and starlet sea anemones are able to use this process. At the University of Miami Rosential School of Marine and Atmospheric Science, a study was conducted which identified special immune cells in cauliflower coral and starlet sea anemones. They used fluorescence-activated cell sorting and microscopy to show how the process of phagocytosis works. Dr. Nikki Traylor-Knowles, a researcher at the university, refers to phagocytosis as the cellular defense mechanism used to eliminate antigens derived from dysregulated or damaged cells and microbial pathogens. This allows researchers to understand the characteristics of a single cell population without the influence of other cells getting in the way. The cauliflower coral clonal fragments used in the experiment were obtained from a single Panamanian genotype. The starlet sea anemones were provided by professors at other universities. In the experiment, researchers exposed the coral and anemones to bacteria, pathogens, and viruses. Once cell populations were isolated by the fluorescence-activated cell sorting, they found that phagocytes were responsible for engulfing the foreign particles, while phagosomes were responsible for destroying invaders and their own damaged cells. In general, the process of phagocytosis in corals and anemones is poorly understood. So why should we care? According to NOAA, coral reefs have a growth rate of 0.3 to 2 centimeters per year for massive corals and up to 10 centimeters per year for branching corals. Because of their slow growth rate, it is impossible to count on regeneration or the growth of new reefs after bleaching events continue to happen. Reefs are severely threatened by pollution, disease, and habitat destruction. When they become damaged, it's harder for them to support the critters that live among the reef. The discovery of phagocytosis in cauliflower corals and starlet sea anemones is a major finding that can help corals stay healthy and is a way that they can protect themselves. The pictures on the slide are from National Geographic, and they show the differences between a healthy reef on the left and an unhealthy reef on the right. So to sum it all up, cauliflower and starlet sea anemones both are endangered and they're both at risk. Phagocytosis is a process where immune cells destroy foreign pathogens. Phagocytes are responsible for engulfing the foreign particles, and phagosomes destroy invaders and their own damaged cells. Both of these species have the ability to use phagocytosis to protect themselves from bacteria and viruses. The study proves that corals have the cellular capability to fight infection and that they have unique cell types that were previously unknown. Additionally, findings from this study can help with the development of diagnostic tools that will eventually be able to assess coral health. This is important because the ongoing climate change crisis is drastically reducing global coral reef biomass and diversity worldwide.